our participants, and I'm especially excited about this session. This is, I think, a really special session because it's all about the future. This is the future of cancer research for small cell lung cancer and the incredible work that is being done by our young investigators right now for small cell lung cancer to move this field forward. Today, we'll be hearing from 3 outstanding researchers who are bringing fresh ideas, innovation, and momentum to small cell lung cancer research. Remember, these were students, graduate students, postdocs who submitted their work. It was a competitive research competition, and they were selected for their research to show you and share with you today. We are joined by Saroshi Pal-Kudri, Carlos Ayala Santos, and Alyssa Cortez, who will all be sharing a snapshot of their work and giving us a look at their research today. They're going to give you an overview of their research, and then what does that mean for you or your loved one going forward, and then the small cell community. Each presenter is going to share for about 10 minutes, and then after all 3 presentations, we'll wrap up with a question and answer session. So as they are talking, again, use those QR codes to submit your questions, and we'll ask them once we're done, whether you're here in the room or you're joining us virtually. Again, this is a really excited, forward-thinking program on the active research in small cell lung cancer that's happening here today. So let's get started now with Saroshi. If you could go ahead and join us here. Thank you. And also, can we have our other 2 presenters go ahead and come up to the stage? Thank you so much. And then they'll go ahead and pop in right after this. Thank you, Saroshi.
Good afternoon, everyone. And thank you, Longevity, for giving us the opportunity to share our work with you all today. Today, I'm going to talk to you about one of the biggest challenges that we face in small cell lung cancer, which is the brain metastasis. That is when the cancer from the lung travels to the brain. This is sometimes really difficult for the patients and their families. However, it has been very hard to study in the laboratory. And our lab has been focusing to develop models which truly mimics what happens in the patients so that we can understand it better and develop better therapies. So this was the slide, sorry. Small brain metastasis in small cell lung cancer is unfortunately very common. And this small cell lung cancer actually has the highest rate of brain metastasis among all different cancer types. However, what's important is that 2/3 of the patients actually develop this after their initial diagnosis and after this treatment has already started. So there is really a window of opportunity where we could either stop it from progressing or have better therapy to treat it. And the difficulty has been that we really do not have a model in the lab where we can actually study this. The models that were being used rarely go to the brain on their own. So if you don't really have a good model, it will be very hard to study. When we are talking about metastasis, it's a complex process where the cells from the your primary tumor needs to go to the bloodstream, travel in the blood, and then go to the distant organ, find their niche there, settle down, adapt, and grow. So in order to study this whole biological process, we really need a complete biological system where we can have all these steps happen. And that is where we used a mouse model, which is called an orthotopic model, meaning here we introduce the cells where they originate. So here we used cells from the patient that are donated by the patients to develop patient-derived models, and then use that to implant the cells in the mouse lung, and then follow the mice to see if it will grow the primary tumor and if it will metastasize on their own to different organ and also in the brain. what we saw in our model is that 100% of the model of our orthotopic model actually developed metastasis in different organs. That also happens in case of patient like liver, kidney, or adrenal, and about 60% of them also metastasize to the patients. To the brain. Importantly, the mice also developed a lot of neurological symptoms that are often seen in the clinic. And we could then follow these mice with different imaging techniques such as MRI, and we could see the lung primary tumor by MRI, which then followed by the development of metastasis in the brain. We could then take the tissue from the lung and the brain and further put them in the slide and look them under the microscope to confirm that they are the actually true cancers and not just some random cells. And this is also the process that is often used by the pathologists and the doctors to confirm disease in patient. So now we truly have a model that mimics the whole biological process that we see in patients and which then we can use to develop or test new drug. So when we dug deeper into how these tumors look in the lung versus in the brain, we saw something changes very significantly. The cells, when they grow from the lung to the brain, they kind of start looking completely different. This is important to study because cancer does not always look the same in different organs. It can depend on where they're originating, but also depends on where they are going to settle down and how they are going to grow there. And what we saw is the genes. So genes are something in the cells that kind of tells the cell what to do, how to grow, what to eat, how to continue to be there. Right? And so we saw a lot of those changes when they grow from the lung to the brain. And the cells in the brain almost started to look like they are like the brain cells or the neurons. They are growing like the neurons, they are eating like the neurons, and they are like developing like the neurons. And this is really important because we cannot assume that the primary tumor where it originates is going to look the same in the different organ. So this gives us the opportunity to actually understand this better and probably develop therapies that will specifically either stop this from happening or target it in the brain. So with that, we now truly have a model that actually mimics all the biological processes that happens in the patient. And the big message that I want to leave with you all today is it's really important to have a good model to understand the biological processes, and to better understanding helps us and gives us the stronger chances to develop new and better therapeutics. And with that, I really want to thank all of you for being here today, all the patients, their families, all the people who organized this, our research collaborators, everybody in the lab, and also the funding agencies without whom none of this will truly really be possible. And thank you all. Thank you so much.
Hello. Okay. So my name is Carlos. I'm an MD PhD student at UT Southwestern, and I'm part of the Povedano Sulfa Lab. And today I'm going to be talking about a little bit of what we do in the lab. But first, let's see. That was a delay click. Oh, yeah. So it's important to understand what a normal cell is before we can understand what a cancerous cell is. And so when we think about normal cells, we have two big categories of genes. Those include tumor suppressor genes and proto-oncogenes. And in simple terms, you can think about tumor suppressor genes as the brakes in your car. When you're driving and you need to stop in a light, you're not going to run the red light, right? You're going to have to stop and press your brakes. And that is important for regulating how the cell grows and proliferates and responds to stimuli. The proto-oncogenes are the ones that tell your cell you need to go, grow, and respond to these stimuli, but you don't want this to go into, you don't want to press the accelerator and go and run because you don't want to get in an accident, right? And so in a normal cell, these two big category of genes need to be regulated tightly in order to avoid that the cell goes haywire and does things that it's not supposed to do. And so in cancer, we don't have that luxury, right? And it's well known that these two big categories of genes are mutated. Either in the tumor suppressors, you lose your brake, so you can't really protect or stop the acceleration or growth. And in the proto-oncogenes, you have mutations that tell the cell you just need to keep going no matter what, and there's no stopping it. And that leads to the development of this disease, or in hindsight, contributes to the development of the disease. It's known that for some models of some cancers, especially non-small cell lung cancers, there are specific mutations that are known to arise in patients that drive the tumor to grow. And those include KRAS mutants, BRAF, or EGFR mutants. And investigators and clinicians have thought that, okay, what if this is straightforward? Why don't we just target this pathway and see if it stops the tumor's growth? And that's exactly what they've done. There are a handful of therapies that are used in clinic right now to treat patients that have these specific alterations. And there are a lot of those that are in preclinical development to treat patients that have these types of mutations and these types of cancers. But what about small cell lung cancer? There is known that small cell lung cancer has loss of tumor suppressor genes, but the presence of targetable pathways like in other types of cancers is not really known. And so what can we bridge the gap to be able to not only understand the biology of small cell lung cancer better, but be able to get to a point where we can develop targeted therapies for patients that suffer from this disease? And so that's where our lab comes in. Our lab uses phenotypic high-throughput chemical screens, which is nothing more that we have a library of compounds, and we're very lucky to be at UT Southwestern that actually has a lot of huge library of compounds that we can test, and we ask the simple question: which one of them are cytotoxic to the small cell lung cancer cell? And what I mean cytotoxic, it simply means that this compound is killing the cell selectively without killing any other tissue types. And so once we know that, we hypothesize that these compounds have a target in which they're engaging, and that target is specifically important for small cell lung cancer to survive. And so now we have the compound, we want to know the target, we work backwards. And so with this, you can start targeting different avenues, and one of them is to understand new biology that is relevant to small cell lung cancer. In our lab, we have found that there's this family of enzymes that convert these compounds into a more toxic chemical, and that toxic chemical engages a target that specifically kills small cell lung cancer cells while sparing others. And so this affects the secretory pathway. We believe it affects the secretory pathway used by small cell lung cancer cells. And so we are, as a lab, are further characterizing this pathway forward. And so having a molecule is really interesting because you can actually develop novel therapies. And so in order to test that before we move into humans, we want to do preclinical development. We want to put these compounds in mice and see if they specifically target small cell lung cancer while sparing your liver, your brain. We need those. We don't want the off-target effects that are associated with the conventional first-line therapies, which is chemotherapy. And so right now, we have this approach. We have many hits in our lab, and we've extensively studied 2 compounds, but we have a lot of other ones that are in the pipeline. Then we really want to know, what are the targets? Are they the same targets? Are they engaging different targets? Are they using different pathways? And all it's for the purpose that we know that they kill small cell lung cancer, but we really do want to know what is specifically getting engaged. Another aspect of my PhD that I'm exploring, and actually our collaborators are just presented and they're in the audience right now, is to study if we can use these high throughput screens to find new targets in chemo-sensitive and chemo-resistant cell lines by asking the question, what is different between these 2 cell lines and what makes them be able to be resistant to therapies? And again, all of this is simply with the whole point of not only discovering new biology, is trying to propel the field forward. We want to find new things that can, even if we're not the ones that are using these compounds to develop new therapies, the information that we are providing to the scientific community will be important for getting to that point, all for the purpose of increasing survivorship for patients that suffer from this disease. And with that, I would like to thank my lab. They're in the first table there. My mentor, Juan Manuel Povedano Sofa. We're a small group, but we're energetic and we're excited for the future and the discoveries that we will uncover. Thank you.
Okay. Hi, everybody. My name's Alyssa Cortez. I'm a third-year PhD candidate at UT Southwestern in the lab of Benjamin Drapkin. It's the same lab as Shirshi, who gave that first BrainMet talk. But today I'm going to talk about something a little bit different. I know this room is, mostly for small cell, but I want to talk to you a little bit about another type of lung cancer that can be very similar to small cell, called large cell neuroendocrine carcinoma, also known as LCNEC. So I wanted to give you guys just a little bit of background about lung cancer and the different types and how it can develop. So Carlos gave a very nice introduction to the different genes that can promote cancer growth for both lung cancer, but for cancer in general. So you have these cancer-causing genes that can turn on, and there's multiple different ways that that can happen. So you can have sort of some type of damage to specific genes, and that's how you sort of get those oncogenes and those tumor suppressors that become mutated. And when these genes turn on, sometimes in the wrong place or at the wrong time, it can cause cancer growth, including the promotion of lung cancer. Now, lung cancer can broadly be distributed across two different categories, being small cell lung cancer and sort of opposingly named non-small cell lung cancer. So non-small cell lung cancer makes up multiple different types of lung cancer. I'm kind of showing you guys three major categories, but there are other ones as well. One being squamous cell carcinoma, another being adenocarcinoma, which is sort of the most common one, and then there's what I study, which is large cell neuroendocrine carcinoma, or LCNEC. Now, LCNEC has a lot of similarities to non-small cell in both its naming, right? The cells are much, much larger than what you see in small cell with some variability. And of course, it also has similarities in sort of those cancer-causing genes that turn on. And one of those examples is RAS. So when RAS is on, it can lead to uncontrolled cell growth. Now, LCNEC is also very similar to small cell. So I mentioned sort of on a cell size, it can be different. One is small, one is large. But also sort of in how they look in general, they can also look very similar to each other. So it can be hard to distinguish small cell from LCNEC. And oftentimes in patients, they can also have a mix of these two tumors where they can have both small and large cell. The other similarity is also with those cancer-causing genes, one of those being ASCL1, which when on at the wrong time, can also promote this uncontrolled cell growth. So in our lab, we found this really interesting, where it seems that LCNEC can have both ASCL1 and RAS on at the same time. And that was sort of unexpected to us. We kind of think of these genes as being very different from each other and being able to promote completely different types of cancer. So as a lab, we really wanted to ask, how is LCNEC able to express both of these genes? How are they turning on at the same time? And based on that, how can we use that information to develop better therapies for patients? Right now for LCNEC, it does not respond quite as well as small cell to chemotherapy or non-small cell to some of its forms of therapies, such as surgery or immunotherapy. So as a lab, we really want to address this and see if we can develop better options. So going into some of those therapeutic options, how do we target some of these cancer-causing genes? So starting first with RAS, which can lead to the uncontrolled cell growth, there's this new drug called daraxonrasib, and it is not quite FDA approved yet, but will probably be very soon. It just finished phase 3 clinical trials in pancreatic cancer. And basically how it works is it basically is able to stop RAS from promoting this uncontrollable cell growth. And in these tumors, they're so used to having this gene that when it's stopped, they do not like it, and it can actually end up in cell death. So that has become a very effective treatment for RAS-driven cancers. And since LCNEC has RAS, it's something we're interested in. Now, on the ASCL1 side of things, it's a little bit more complicated. Right now, we don't have a therapy that directly is able to target ASCL1, But we can take advantage of things that ASCL1 itself is able to turn on. So for example, ASCL1 is able to turn on DLL3. And for anyone in this room who's familiar with tarlatamab, this is how tarlatamab takes advantage of that ASCL1 phenotype, where you see your cancer cell has DLL3, and we can treat with tarlatamab, which is currently FDA approved for treatment in small cells, sort of as a second therapy after chemo. And sort of one half of tarlatamab recognizes the DLL3 in the cancer cell, and the other half is able to recognize an immune cell. And it basically points out to the immune system that there is a cancer cell present, it's doing harm, and it needs to be destroyed. So the immune cell is actually able to kill the cancer cell. And since LCNEC has both of these cancer-causing genes, oftentimes, both RAS and ASCL1, we really wanted to develop strategies of how to combat both oncogenes. So what I'm showing here on the right is just a little bit of data from our lab where we took a human LCNEC tumor, so this was donated from a patient, we made it into a model, and we were able to grow it and then treat with these different compounds in mice and asked what happened to the tumor. So what you're looking at on the x-axis is time in weeks, and on the y-axis, you can kind of see the starting size of the tumor. Anything that goes up grew, and anything goes down shrinks. So in the black line, these are untreated mice, meaning they got no drugs, no medicine, and you can see how quickly those tumors grow for this model. And then sort of in the red line is our combination treated. So these mice were treated with daraxonrasib and tarlatamab, and you can see that they very nicely decreased. So that was something we were really excited about. And we've also been very well aware of anytime you're talking about combining drugs, making sure that they have different side effects so that you don't have some adverse reaction. And so far, these mice have done very well on this treatment. So this is something that we're hoping to do a little more research on. This is sort of, we just got this data, so I'm really excited that I got to share it with you today. So we're going to do a little bit more work on it, and then we hope that this will become an effective treatment in patients, is sort of the big picture of where we'd like to go. And that's really all I had for you. So with that, I just want to thank, there's a lot of people who contributed to this work. So to thank my lab, as well as other labs we work with at UT Southwestern. And of course, I would love to thank the organizers for the opportunity to speak to you all today. As a researcher, I don't really get to ever speak to patients, so this is exciting for me. And I'd like to thank you all for coming, patients, caregivers. So thank you.
Absolutely fabulous. Can we give them one more round of applause? Thank you all for what you do and your research. I am so grateful. I know so many of our smashers here are grateful. And thank you for moving the needle forward by advancing research and putting this at the forefront to really help our patients and loved ones have new options, understand the biology, and make small cell a thing of the past. So thank you again. I'd love to open this up for any questions if you have in the chat. Anne will kick us off.
Yeah, the first question in the chat, I actually think both of these are for Shiroshi, and when small cell metastasizes to the brain and takes on the attributes of brain cells, is it also true when it moves to other organs?
So we are currently looking into that. So for example, it's very common in many of the cancers when they kind of change to some extent to specifically adapt to the microenvironment of the distant organ where they're growing. But for brain, the cells look completely different. The neurons are really truly different for any of the cells that we have in any of the other organs in the body. And the metabolism, meaning how they eat and how they grow, is very different. The immune environment is much more restricted to any other organs that's in the body. So that difference is really striking. But yes, to be straight, like in any other organ that they find the niche, they do need to change to some extent to adapt to that organ and grow there.
And a follow-up to that question was that did any of the mice's cancer spread to their other organs or bones rather than to the brain?
Yes, so almost for 100% of the mice, it did go to their liver. In some cases, it went to adrenal glands or kidneys. In female mice, it also went to ovary. It also went to the mediastinal lymph node, the lymph node that are closer to the lung, which is also often seen in case of patients. And those are the main organs where you found this metastasis.
Please put your questions in the app if you have other questions.
Sure. And I'll keep picking your brains. I mean, we have 3 very bright stars as young investigators here, gracious to share their work, and love to ask each of you some questions. So, Alyssa, I'll start with you, and we'll work this way. So, one of the main areas, as mentioned by Shiroshi, is areas in the brain. Do you think that your regimen for large cell neuroendocrine might be effective in the brain? Are you guys studying that?
Yeah, so that's a good question. So we haven't gotten a chance to sort of study the metastatic potential of LCNEC yet. Sort of this idea that it has these 2 genes is kind of new. So we're starting sort of in a simple model where we grow it in the mice and it's not able to metastasize in the location that we're measuring. But it is something we've thought about, is using that orthotopic model that Shiroshi's developed to see where they go and how these different treatments are able to impact that. In patients, we have seen it does most patients that when they are diagnosed with LCNEC, it is already typically undergoing metastasis. So that is a big thing for LCNEC as well.
Yeah, so I think an important unmet need, and I'm really hopeful for your combination studies to highlight that efficacy, so you'll have to update us. Carlos, tell me more about really kind of your research. What are you hoping in this high throughput screen? When you find a hit, a new drug, how do we take it from the lab, right? You and your team, how do we take it from the lab and get that to trials? How do we, what's that pathway look like? How do we get this where we can eventually get it from a bench to a bedside?
Hello? Okay. So drug development is complicated. So we have a collaboration with chemists at UT Southwestern because this library, we get these hits, but then we try to make what is known as structural activity relationships. So we modify the compounds in a way to really see how it affects this activity or makes it more potent. That's why we need to move it to, once we have these compounds, we want to know what the target is. And once we characterize that pathway, we want to try to move it then again into mouse models to see if it truly does specifically target the tumor while sparing the other areas of the mice in general. That's the first step before pushing it into preclinical development in humans. If it works in mice, then we can scale it up. That's expensive, but we have these lead compounds that can provide some good information for the development of a targeted therapy and hopefully a development of a targeted therapy in the future, and then that can be pushed into human subjects and see if it works.
Yeah, I think what you're highlighting, Carlos, here is it is complex, right? It's a lot of steps. And safety is of the utmost importance, and making sure kind of steps along the way have been taken. We're checking kind of in a systematic fashion before we get there, right? Before we get to patients. But that sounds like the ultimate goal is to get these exciting drugs as they've been optimized, understood, to get them to our smashers. And then Shiroshi, with your brain metastases model, what are some of the challenges that you face day to day in the lab as you're studying these, and what are some questions that you wish you had answers to? What are some things that, as you're doing this as a researcher, that you've seen that you might want answered going forward?
So I think the biggest challenge that I would like to address is how to stop them from going to the brain at the first place. because as you see, 2/3 of the patients don't really have a brain met when the disease started or the disease is diagnosed. So the best option will be if we can understand the cells that are in the bloodstream and then target them there before they can reach the brain and find a place there. And of course, if they're there, then we have to really make sure, because brain is all the organs are important, but it's also like we do not want to damage any of the surrounding areas. So we really want to hit the tumor where it's there and not have any effect in any of the surrounding cells that we need to have our quality of life. The biggest challenge in the labs have been truly developing this model and actually able to find a model that will go to the brain on its own. Because you do really need to have this biology of it going to the brain by their own that happens in the patient versus if you inject the cells directly in the brain and study that. That does not really replicate what you see in the patients.
Thank you for highlighting that, Shiroshi, and the challenges that you face and the things you're thinking about. And for our smashers, really, this is something called cancer interception. How do we stop or intercept it from going to the brain? And I love that. That's such a perfect focus of how do we even stop the formation and try to be as proactive as possible. So thank you. Do we have any other additional questions? Okay. But again, please, let's give a congratulations and an applause to all our investigators. Again, thank you so much from the heart of all of us of what you're doing. And these are the faces and the patients that are going to benefit from your work. And so thank you. Thank you for making small cell so important in your work. So thank you all. Thank you for listening.